So you'll recall last time we had spent quite a bit of time talking about influenza, and I think we had just started to introduce rabies virus. Does that sound about right? Okay.
Yeah.
So we just want to continue with our RNA viruses. So here we see an electron micrograph of the rabies virus. It's plasma, it's bullet shaped. It has an envelope, which is very good because that means it's not going to remain infectious outside of its host for very long. And then we see the little glycoprotein spikes of the envelope that will permit the virus to attach to host cells and infect them. Now, here in the United States, because of vaccination campaigns, rabies is not the killer that it is elsewhere in the world. But we want to be aware that worldwide, there are many, many, many deaths worldwide from rabies. And the majority of these deaths occur in areas where there aren't good vaccination campaigns, for example, for dogs and cats. And in addition, it's important to remember that even if you're working in medicine here in the United States, travel history will be very important for you because, especially here in California, people love to travel. A lot of us have families that live in different parts of the world, so there's a lot of traveling, a lot of immigration. So it is important for you all to remember that worldwide rabies still is a considerable public health issue. Okay, so with regard to reservoir hosts, important reservoir hosts for rabies, we can see that skunks, fox, and raccoons are significant reservoirs of rabies in different parts of the country. And you see here in California, they have us highlighted with skunks being an important reservoir. But it's actually unfortunate in this cartoon because bats throughout the United States are important reservoirs of rabies. And especially here in California, bats are important reservoirs of the rabies virus. Some biologists believe the rabies virus has been co-evolving with bats for long periods of time. And the reason is bats may become infected and may be asymptomatic. And it's also really interesting, they usually don't develop an antibody response to the virus. So we know that normally bats are nocturnal. They're important insect eaters. They play a really important role in ecology. Here in California, they are very helpful because they eat lots and lots of mosquitoes that can transfer pathogens. So some people have suggested we should just wipe out all the bats since they harbor the rabies virus, and ecologically that would be a disaster. And again, we want to remember they help control arthropods that could transfer pathogens to humans. So again, we want to remember bats is an important reservoir. So certainly if you were to see a bat on the ground during daylight hours and it's not trying to fly away from you, you probably want to stay away from it because it is possible that is it possibly could have rabies. Carmen Percala, who is our biology coordinator, was bitten by a bat. She was working with Professor Dave Wyatt, and he's a bat expert. She was bitten by a bat, and because she didn't want the bat to be destroyed for rabies pathology, she underwent the post-exposure rabies prophylaxis. So Carmen has had experience. The post-exposure prophylaxis involves vaccination with the rabies vaccine. You also receive neutralizing antibodies, gamma globulins against the rabies virus. And the idea is that these antibodies will bind to the surface of the rabies virus so it can't invade your neurons, your nerve cells. And then in addition, usually a tetanus vaccine is given. Professor Virginia Meyer, who some of you may know, who also teaches microbiology, she was working with Professor Dave Wyatt. Oh, I'm seeing the common lake here. She was working with Professor Dave Wyatt, who also does studies on ringtails up in the Yuba Buttes, the Sutter Buttes. And she was bitten by a ringtail, so she too went through the post-exposure rabies prophylaxis. So we have two experts on post-exposure prophylaxis here in our department. Now, the reason that folks can be vaccinated for rabies after exposure, it's thought that the rabies virus first replicates maybe in the muscle cells near the bite site, and it takes a while for them before they'll have made the neurons. So the normal infection with the rabies virus, it triggers really low and slow antibody production. Like if we were to be bit today, we eventually make some antibodies against the virus, but there wouldn't be enough and in time to prevent the virus from invading our nerve cells, our neurons. In contrast, the vaccine triggers fast, strong antibody production. So this is why you can vaccinate a person after exposure, get them to start producing those wonderful neutralizing antibodies, which will block attachment of the virus before it can invade the central nervous system. Once the virus is in the central nervous system, the vaccine is no longer of any benefit. So you have to start that post-exposure prophylaxis rapidly after you've been bitten. This is just to demonstrate why one of the options when somebody is bitten by an animal that might be rabid, one option is to sacrifice the animal. That means to kill the animal and take a brain biopsy. And the reason for this is that in about 80% of animals, except for bats, bats don't tend to do this, in about 80% of animals, those that are infected in the central nervous system with the rabies virus, you can actually see these little negri bodies. Negri bodies that accumulate in the neurons, and these are accumulations of the rabies virus that's actually replicating. So this is why one of the, I guess, older historical ways to diagnose rabies was to kill the animal, take a brain biopsy, stain it, and look for these negri bodies. But we do have to remember in 20% of the cases, rabid animals won't have the negri bodies. So if you find them, they say it's pathognomonic for rabies, so you know it's rabies for sure, but you have to remember that in 20%, the rabid animals, you might not see the negri bodies. Now, from the, once the rabies virus invades the nervous system, it reaches the central nervous system, it replicates in the brain, and then using neurons again, nerve cells again, it travels back down to the salivary glands. And in the salivary glands, it will be shed, and that's how, in animals at least, rabid animals shedding rabies virus in the saliva, if they bite another animal or a person, that's how the rabies virus will be transferred. Now, the rabies, the original rabies vaccine was developed by Louis Pasteur. I mean, Pasteur was just this phenomenal person, you guys. I cannot believe how much he did. So Pasteur had been working with a bacterium that caused a disease in birds called fowl cholera. So fowl as in not fowl, F-O-U-L, but fowl as in the birds cholera. And this is a bacterium called Pasteurella multocida. Pasteurella multocida. And Pasteur's experiment with this bacterium is that he took the bacterium and he would pass it sequentially in the lab on auger plates, just like you guys know how to do. So he would isolate a virulent strain from a bird that had died. He would plate the organism on auger plates, and then sequentially he would transfer them to plates after a week or two. And so he kept doing this in the lab, passing them serially. And what he discovered was after several weeks of passing the bacterium on auger plates, it had become attenuated. So we'll say serial passage in lab, so not growing it in animals. So what he discovered is it caused decreased virulence. And whenever we have a pathogen with decreased virulence, we call it attenuated. It's almost like you weakened it. Attenuated. And what he did then was he used this attenuated Pasteurella to inoculate healthy birds. And what he discovered was the healthy birds didn't get sick. And in addition, when he took those birds that he vaccinated and challenged them with virulent Pasteurella multocida, they also didn't get sick. So he showed that by passing these pathogens in the lab, you can attenuate them, weaken them, and then you can use them as a vaccine to protect animals against highly virulent pathogens. Well, the reason I brought this up, you're probably wondering, why the heck do we care about fowl cholera? Well, it was this experiment that triggered Pasteur to attempt the same process with the rabies virus. What he did was that he took brain from an animal that had died from rabies, maybe it was a rabid dog, and he took brain material, ground it up, and then he inoculated rabbits. And he inoculated rabbits in the brain, so intracerebral inoculation. Wow. And in rabbits, when they get inoculated in the brain, the progression of the disease is really fast. Like they're going to develop signs and symptoms in 3 to 4 days. So what Pasteur would then do, once he knew that the rabbits were infected, he would kill them, take brain material, and inoculate another set of rabbits. So you guys know as a virus, you couldn't use agar plates, right? Like you did for the Pasteurella. He had to use animals. So he serially, reminds me of eating, I'm hungry. Serial passage through the rabbits. And what he was hoping is the same phenomenon would occur, that he would get what? Right, decrease in virulence of this lab-passed rabies virus, that he would have an attenuated virus. And so what he did is that he used the attenuated rabies virus and he vaccinated dogs. And then what he did is he challenged the dogs with highly virulent rabies virus, freshly removed from a rabid animal. And what he discovered was the dogs were protected, right? So that was a huge step. Now, unlike Jenner in the 1700s, and we're going to be talking about Edward Jenner, who developed the smallpox vaccine, Pasteur had the ethics not to attempt this in a human model. I mean, he didn't want to inoculate somebody with his rabies vaccine and then challenge them with the rabies virus, right? Because rabies is about 100% fatal. So he, this was great progress, but he didn't want to try it on humans. But what happened then was that word of his work got out, the public knew about it, and 2 families presented their sons that had been bitten by rabid animals. And everybody knew, if your kid gets bitten by a rabid animal, they're going to die. So these families in desperation came to Pasteur and said, please, can't you do something to help our kids? And so Pasteur reluctantly said, well, I'll try this vaccine, can't hurt. And so he gave many inoculations of the vaccine over time. And the incredible thing was these little boys were saved. It was just, it was like a miracle. It was like, good heavens, that's just absolutely wild. So the vaccine, and I'm sorry you guys, the vaccine was made by taking the spinal cords out of the rabbits that had been serially inoculated. He would dry the spinal cord and grind it up. An idea is the attenuated rabies vaccine was present in the spinal cord. He would use that as a vaccine. So to save these little boys, they received 14 injections, but they were indeed saved. And there's even, and this might be folklore, you guys, but I love stories like this. So one of the little boys, when he grew up, he was so thankful to Pasteur. He worked at the Pasteur Institute, this wonderful research facility that was developed in Paris. And part of the folklore is in World War II, when the Nazis invaded Paris, this grown survivor of rabies that Pasteur protected, was blocking the doors of the Pasteur Institute, not wanting the enemy to pass. And I just oh, that's such drama. Great stuff. Okay. So what an incredible development, this rabies vaccine. So subsequently, what was discovered is that if you use vaccines made with nervous tissue in it, sometimes it'll trigger really severe adverse side effects in people. Maybe in some people, if you inject spinal cord, for example, it'll actually cause an inflammation of their own nervous system. Maybe it triggers an autoimmune response. So as technology developed, what they started using for the human rabies vaccine is they started growing it in embryonated duck eggs. Just like the chicken eggs you were talking about, right? But the problem with that duck embryo rabies vaccine is that it doesn't trigger really good antibody production. So, for example, in folks that were undergoing post-exposure therapy, sometimes they had to get 20 injections, you know, to try to, yeah, right. So, so subsequently what they did, and this must have been like around the late 70s, early 90s, they developed a tissue culture cell line made from human diploid cells. And that triggers a much stronger antibody response. So this human diploid cell vaccine is, I think, the most common vaccine used here in the United States. It's used for folks that would be at high risk for rabies. So, for example, like if you go to vet school, you're going to be vaccinated against rabies. Folks, obviously, that are working with wild animals like bats, like Dave White's been vaccinated, Carmen's been vaccinated. Folks working in research labs, you'll get vaccinated prophylactically to prevent, you know, infection to start with. But then again, we do want to remember, it's incredible, with rabies you can get vaccinated after exposure and still generate protection. Okay, so this is just to show us that because the virus is replicating in the brain, it causes behavioral changes. In some animals, it makes them very aggressive. And there's a lot of interest in microbiology, how microbes affect our behavior, right? And so you could argue that this is, this behavioral change, making the infected animal aggressive, where they're going to go around trying to bite, is brilliant, you know, strategy on the virus's part, because this is how the virus will be spread to new hosts. And the horrific thing is in humans, you can get, you know, horrible neurological changes. This poor person is in the later stages where he's undergoing spasms, muscle spasms. And in most cases in humans, like we said, once the virus invades the central nervous system, usually it's 99.9% fatal, and it's usually fatality from respiratory arrest, so you're going to suffocate. Yeah, a little horrible way to go. Yeah. So there was one case, and I forgot to check the details, you guys, before we came. That within the last few years, there's been one case where a young woman was infected, the virus did invade the central nervous system, and what they did, they put her in a drug-induced coma and just kept her that way, and she survived. So let me try to research that and just give you the reference on that, because that is significant. She made it. She survived. Another important thing to note, you guys, is that often, especially with bat rabies, people have no memory that they were ever bitten. So remember that. Some people can become infected, especially with bat rabies, and they're like, I was never bitten by a bat. And there's a couple of explanations. One might be if, like, they're camping or in a little cabin and a bat gets in the house. You know, there's some people say, is it possible they were bitten during the night and they didn't know it? I don't know. That sounds kind of far-fetched to me. But another concern is, I think we had mentioned, if you like to explore caves, since the rabies virus is shed in bat saliva, and bats as social animals are always grooming one another, okay, in caves, in these enclosed spaces, where bats live, you can have aerosolized rabies virus, so it might be through like inhalation even that you get infected. So do remember, sometimes people will not remember they've been bitten by an animal, and yet they do indeed have rabies. There has been one report, this is really sad, 2004 here in the United States, where somebody died of this neurological disease, and they didn't know what the heck it was, and the family agreed to have the kidneys and liver donated to 3 separate recipients, and within, let's see, it was like 6 to 8 weeks afterwards, all 3 of the recipients died, because the person, the donor, had died of rabies and nobody knew it. And so the neurons in the donated tissues had the rabies virus, so the poor recipients all ended up dying. So, you know, bizarre neurological diseases, probably, now that, we know we're going to talk about put-on diseases, now we know rabies, probably if somebody dies of a neurological disease that never gets diagnosed, probably not a good idea to use them as tissue or organ donors, because you don't know what caused that. Yeah.
You don't check that before.
Well, I think the reason is rabies is so, here in the United States, the numbers of rabies cases, human rabies cases, is so low. People would like, maybe if somebody might have suggested it, and they're like, oh, come on, don't be ridiculous. And so it just wasn't high enough on the folks' radar. But again, you guys, nowadays, especially after we talk about prion diseases, any kind of neurological, severe neurological disorder that ends up in the patient dying, I don't think that's a good donor. You know, I said, if you don't know why they died and they have severe neurological disease, does it make sense to use them, you know, bless their hearts, that they, their family would offer their tissues and organs for donation? Probably not a good idea, because you might be passing whatever the agent is on to the recipient. So that's kind of a heartbreak. Okay, so we're going to leave rabies, you guys, and we're going to just so quickly touch on one arbovirus that's made its appearance here in California and in the United States within the last few years. And the RNA virus we're going to discuss is West Nile virus. And West Nile virus belongs to a group of viruses called Arboviruses. And Arboviruses stands for arthropod-borne, so Arboviruses. Viruses that are spread by arthropods, just as we learned in lab. And as you all learned in lab, mosquitoes are wonderful arthropod vectors. They're blood feeders. So when they take a blood meal, if a pathogen is present in the bloodstream, they ingest the pathogen. And then subsequently, when they bite a new host, they will inject the pathogen. And often this is through injection of saliva. Blood-sucking arthropods, often when they feed on a host, they have to inoculate a little bit of anticoagulant. Otherwise, the blood might clot and clog up their mouth part. So with mosquitoes, they have like this long, hollow mouth part which pierces the blood vessel. It's like a little hypodermic syringe. They suck up the blood. But you can imagine how narrow it is. So if there was any blood clots, you know, then they couldn't feed anymore. So they inject a little bit of anticoagulant. And some blood-feeding insects, they'll also inoculate a little bit of local painkillers, so you're not aware you're being bitten. So it's when they inoculate us with that saliva, that anticoagulant, often the pathogen is transferred. So just really quickly, you guys, as I mentioned, we're just going to talk about one arbovirus. Some of the other ones that are probably famous worldwide, yellow fever is an arbovirus carried by mosquitoes. Dengue virus, also an up-and-coming arbovirus. EEE stands for Eastern Equine Encephalitis virus. So this is kind of an overview of the transmission of many of these arboviruses. So we'll just do our own here for West Nile virus. West Nile virus, as far as we know, was first introduced to North America in the late 1990s, around 1999. So here in North America, it is considered an emerging infectious disease, a new infectious disease. So in the United States and the U.S., we think it was first detected in around 1999. And it was really interesting because in New York City, NYC in New York City, they had two unusual disease outbreaks going on at the same time. They had this febrile illness. Febrile means fever. And nobody could figure out what was causing it. And then at the same time, people were noticing that crows were dying or acting odd, like they're having neurological problems. And because so often we keep, human medicine and veterinary medicine separate, as if we are part of the animal kingdom, right? At first, people didn't get the connections that maybe this could be the same disease. And my understanding was it was actually a veterinarian that suggested, hey, this disease that's going on in the humans and this disease that seems to be killing a lot of birds, what if it's the same disease? And it turned out they were right. Okay, so this was a new virus that had been imported. It is present in northern Africa, in the Middle East, and in Europe. It's been around probably since like the late 1930s, but we've never had it. Okay? And it was eventually identified as this West Nile virus, named after the location of some of the first cases that were described in northern Africa. So it was called the West Nile virus. And the natural cycle for West Nile virus, so W and V, the kind of the natural it evolved as a virus of birds. And so in the natural cycle, infected birds are bitten by mosquitoes. Some folks think this is a great name. They describe mosquitoes as flying hypodermic syringes, so they are out taking blood and infecting other places. And then the natural cycle is an infected bird is bit by a mosquito. The mosquito takes up the virus. The mosquito bites another bird, injects the virus. That bird becomes infected. That bird's bitten by another mosquito. And then the cycle just goes around and around and around. Now, some birds can carry the virus asymptomatically. And usually when that happens, you think, oh, maybe that was like the original host, the virus of the bird. They've been co-evolving for many generations. The individuals, birds, that are highly susceptible, they end up dying. They don't have as many offspring. The birds that are naturally more resistant, they survive the infection. They pass on the genetic information for that resistant to their offspring. So you have natural selection for resistance in certain bird populations. Now, not all birds are that resistant. Like we said, and crows are really susceptible to serious disease. Scrub jays, blue jays, magpies, those birds are really susceptible to serious disease. So they haven't been co-evolving as long. Now you might say, well, okay, so where does the people come in here? We see where the birds are involved. Well, it turns out some mosquitoes are not fussy eaters. Some mosquitoes are very specific. They'll only bite one kind of host, right? But some mosquitoes, for example, Culex mosquitoes that we have all around Sacramento, they are not fussy eaters. So if they get hungry and a bird's not around, but you are, they're just as happy biting you. So they'll bite you, they'll bite humans, they'll bite almost anything that moves. But another host that we'll talk about, do any of you all have horses? Anybody have horses here? Usually, most semesters, one or two folks have horses, and that's why we bring up horses. So Culex mosquitoes, which, as we said here in Sacramento, Northern California, we got lots of Culex mosquitoes. If they can't find a bird, they're just as happy biting humans or just as happy biting horses. And indeed, the West Nile virus can be transferred to humans and horses. Now, the impacts are different. In humans, if you have a good immune system, probably the majority of us, all we would experience would be like a flu-like illness. And we're going to discover that most viruses cause flu-like illnesses. That's what makes it so hard. So most of us, in fact, maybe a lot of us have already been infected with West Nile virus. We just don't know it. Okay. So if you have a good, strong immune system, maybe flu-like kind of, maybe a little bit of fever, but not that big a deal. But the problem is we're going to see over and over again, folks with compromised immune systems, and there's probably some genetic aspects to this. Even folks that have normal immune systems but maybe are genetically predisposed. Unfortunately, some of these, a few people will experience neuroinvasive disease, where the virus invades the central nervous system, and in some cases can actually kill people, and in some cases can cause permanent neurological damage. So the problem is, it's just a tiny percentage of people that will experience severe disease, but can be devastating if you're one of those people. Now in horses are incredibly susceptible. So with horses, very often, it's frequently neuroinvasive, and it's caused the death of many, many horses. So if you are a horse owner, you're probably aware West Nile virus can kill your horses. Now, we're going to shift perspectives, you guys, and we're going to pretend that we're taking the perspective of the West Nile virus. If you're the West Nile virus, you don't want to end up in a human, nor do you want to end up in a horse, because from the virus point of view, these are what we call dead-end hosts. Okay, so a dead-end host means, from the pathogen's perspective, you're going to die with that host. And the reason is that in humans and horses, the virus numbers in human blood and horse blood never gets high enough to infect what vector? mosquitoes, right? So if you think about it, you guys, when mosquitoes take a blood meal, I mean, for them, it's a big amount of blood, right? But if you think about it, how much blood we have, and then think about, okay, this tiny, tiny, tiny aliquot of blood the mosquitoes suck up, you're going to have a pretty high virus concentration or virus titer in your blood to infect mosquitoes, right? So do you think we're going to get that high concentration of West Nile virus in human or horse blood? No. And that's why they're considered dead end hosts for the virus. That means the virus, once it gets into humans or horses, it will not be picked up by a mosquito and spread to a new host. So this is the end of the line for the West Nile virus in humans or in horses. And we could also argue, well, for most horses, it's the dead end for them too, and for a few humans, it will also cause death. So kind of almost a double meaning here. Okay. Now, so this is important when West Nile virus finally was brought to California, and we think it was brought from the East Coast to California by migratory birds. A lot of people got so upset, they're like, oh my gosh, my neighbor has horses, and those horses are going to get West Nile virus, and then we're going to get West Nile virus. So people are, arguing with their neighbors, you should get rid of your horses, blah, blah, blah. So just remember, you guys, horses can act as a reservoir. They can die from the virus, but they're not going to act as a reservoir to infect mosquitoes that would infect you. Okay, so that's something to remember. Now, the good news is they do have an equine vaccine. Equine is just a fancy name for horses. And when it first came out, your colleagues were telling me it was really expensive. And often, I think you had to get at least 2 of the vaccines. They said it was incredibly expensive. But subsequently, some of your colleagues have said it's become much more affordable. But it is, if you have horses here in California, you really probably should get the vaccine, bite the bullet, bite the bite. Okay, all right. But of interest, there's no vaccine for humans. And it might be, I don't know, I'm being cynical, you guys, it might be since a relatively small number of people get seriously ill, and developing human vaccines is really expensive. I'm just wondering if the vaccine companies feel like, there's not going to be enough profit. The other thing is we mentioned, probably a lot of us have already been infected, and the model is that if you're infected, you'll have lifelong immunity. That's the model. Of course, there's always flaws. But it might be vaccine companies figure, it's not going to be worth it for us. But I always think that's interesting. There's a veterinary vaccine and not a human vaccine. If any of you end up working in zoos, often what they'll do is they'll use the equine vaccine to vaccinate zoo animals, because a lot of them are highly susceptible to West Nile virus too. Yeah, so big things with these mosquito-borne diseases is mosquito control is important, and then just what we can do. You've probably heard from the mosquito control district, they ask you to go around your house and dump containers that have standing water. Female mosquitoes have to lay their eggs in water. And so if you can get rid of standing water, like in old tires is a big one, like flower pots, that might have water in them. I know in our pets' water bowls in the backyard, I went out there last weekend, and here's all these mosquito larvae. I'm like, cats. In some mosquitoes will lay their eggs in holes in trees, where water will collect. So you might even want to check your trees to see if you've got little collections of water. There could be mosquito larvae in there. And then they say, of course, to have screens on your windows. And then they say avoid going out, in the evening or early morning when mosquitoes are biting. I'm like, this is California. This is why we live here, so we can be out doing all kinds of crazy things at night and early morning. So, but maybe mosquito repellent, would help a little bit there. So there are things we can do to control the vectors. Oh, and they said this has been a huge issue. Another one of the tragic, consequences of the economic downturn. A lot of folks that have lost their homes, and have had to leave their homes abandoned. Some of those homes have pools, and so they aren't, maintaining the pools. And pools, if they aren't kept properly chlorinated, become wonderful breeding grounds for mosquitoes. So they say the mosquito control abatement, they fly, they do aerial flights looking for green pools, because green pools suggests algae is not being taken care of, and could be a huge source of mosquitoes. So we're not, oh, and this is just a little bit probably from lab. You guys remember that with regard to mosquitoes, usually the males don't feed on blood. Males are actually important pollinators. They feed on nectar, so they help pollinate plants, which is becoming more important as our honeybees are disappearing. So only the females are blood feeders, and of course we can make all kinds of great jokes about that. So only the females are the blood suckers. Okay. So in our house, my husband was trained in entomology, which is a study of insects. So you know how nerdy we get, right? So when mosquitoes are flying around, we're like, sex it, sex it. If it's a male, we let it go, and if it's a female, we're like, whack. Okay. This is just to show that with these arthropod-borne diseases, if you track the arthropod vector numbers, and usually it's associated with warm temperatures. So here, May, July, now we're getting into fall. We're dropping off in wintertime. What they're trying to show you here is that the number of cases of West Nile virus are temperature related. So as the temperature increases, the number of West Nile virus cases increase. And this is because this is when the mosquitoes are hatching. So it's during the warm months, so spring through summer, that we get the greatest number of mosquitoes hatching. We have the greatest number of mosquitoes out feeding and transferring West Nile virus. So the temperature correlates with increased number of mosquitoes. Increased number of mosquitoes means we're going to have an increased number of West Nile cases. So that's why mosquito control is so important. Unfortunately, you guys, we're not going to talk about dengue virus. It's another really important arbovirus. It is spreading worldwide. And you just in a nutshell, I'll just give it this, because again, it's important. It's like, just shut up about these things. Okay, so the importance of dengue virus is there's 4 different serotypes. Now, prior to like World War II, places that had dengue virus, they only had one serotype, right? If you're bitten and the first time you get infected with dengue virus, you could get really sick, right? It's not that it's a fun disease. It's often called breakbone fever because people say you feel like your bones are breaking, it's so painful, right? But the first infection, usually you won't die with the first infection. What puts you at really high risk of another type of dengue viral infection called dengue hemorrhagic fever. Hemorrhagic means to bleed out. Is if you subsequently get bit with a different serotype. Now, prior to World War II, this wasn't that big a deal. People didn't move around the world so rapidly. But World War II came and people were moving all over the world. And then what happened? The Korean War came and people were moving all over the world. And then what happened? The Vietnam War came and people were moving all over the world. So as a consequence of that, dengue viruses, the different serotypes got spread and mixed up all over the world. So the problem now is that especially if you're a traveler, if you have like family, like we have family in Malaysia in the tropics, if you're a traveler and you get bit with one serotype of dengue, you know, and if you're a traveler and you're going and visiting to other areas, you could get bit and infected with a second type. And then you're at very high risk for dengue hemorrhagic fever, and that's a killer. So you guys, just to give you, you know, sometimes when it's like your family or friends, so like I said, my husband comes from Malaysia, so his sister-in-law has been infected, his niece has been infected, and in that same neighborhood, a 5-year-old child died from it. So I mean, it's there, right? It's out there. It has caused outbreaks in Hawaii, it's caused outbreaks in Florida, and cases in Texas as well, okay? And the concern is with the climate change and some areas becoming warmer, the fear is that the vectors are going to, their range will increase. So the concern is if truly the climate gets warmer, the vectors for dengue could move northwards. So it could be a virus that here in California we may have to add to West Nile virus as another significant mosquito-borne virus. And again, because so many people travel here in California, potentially that would increase the risk of folks developing the dengue hemorrhagic fever. And another reason why travel history is so important, you guys that are going into medicine, you have to get that travel history. You have to know where your patients have been Just because we don't have it here in California doesn't mean they didn't pick it up on their travels. Okay. Oh, cute. So with that happy note, one of my friends who used to work in the department, she goes, so what new depressing thing are you going to teach your students today? She goes, I wouldn't be able to survive those lectures. I'd be so depressed by the time I came out. But hopefully, in understanding the natural history of some of these pathogens, you guys, we cannot be afraid of them. We can understand how we can control them, right? So hopefully it's not all depressing, like, oh, we're all doomed to be infected and hurt by these pathogens. We're learning to respect them, right? We're learning ways to avoid being infected, and we're also learning how understanding the natural cycles help people find new drugs. And boy, the greatest example I think of this is looking at HIV, the human immunodeficiency virus, which is going to be our next topic. We're